Welcome back, students. Am I audible and visible to all?
Yes, sir.
And hands are also visible.
Yes, sir.
Slides are also visible.
Yes, sir.
Yes, sir.
Okay, so we can wait for a few more minutes. So meanwhile, all of you will join. Then we'll start. Okay, so Rupal want to say something? Yes, Rupal.
Yes, sir. Sir, actually, I was absent on last Wednesday, so you haven't checked my paper yet. Midterm lab.
Okay, so that I will do and let you know the marks.
Okay, sir.
So that I want to do now on here. Okay, so I will do that and I'll post you the marks.
Okay, sir.
So we can start now. So you are 21 now, so 2 are absent, I think, and those who are coming to join now, they will be marked absent. So one is among them is a Kshitij. So I will extract the attendance list at the last. So I will keep the marking of 12:25. So who is going to join after 12:25 will be absent. Okay. In our previous classes, we have talked about different types of the vectors. So different property is going to be how you develop these vectors and how you reach to the individual vector, what component from where you got it. Now the next thing is the expression. So as a term expression is there, it means when we are cloning a nucleotide sequence, the nucleotide sequence is going to be your gene or your gene of interest. Now we want to see its product. It should express. So those vectors which assist you in the expression of the gene, they are going to be called as the expression vector. Okay, so whenever you're calling the expression vector, so simply thing is that we need all the machinery which is going to play a role in the transcription. So transcription of a gene, because DNA is a genetic material, it contains the information, but this information can't be translated. So transcription in the translation, until unless it is being translated, we cannot say that your gene is expressed. Gene is expressed only if you are having a product of it. So product of a gene is going to be a protein. So that protein is when you say that this is going to be expressed. Or even if you say it's not a protein and it is RNA, it's the RNA. So it is also a product of a gene, depending on what your which is going to be. So thing is that when you want to express a gene, you need all the machinery should be present there so that you can actually get the product of the gene. Okay, so if a foreign gene simply ligated to a standard vector and that clone in the equili, it is very unlikely that significant amount of recombinant protein will be synthesized. So recombinant protein is going to synthesize recombinant protein. So for the recombinant protein, what you need, how to express it. So for that you need to go back to the machinery like in your, I think second year that you have studied about, like operons. So do you know about the operon? Do you know about the operon?
Yes, sir.
So which operon you studied?
Lab
That's
open.
lack Operon.
Okay, so how you define the operon?
Operon.
How you define an operon? So what do you mean by operon? Okay, but I won't define it. Yes, Ishit.
Genes.
Huh?
Sir, it is a set of genes. Genes, cluster of genes.
Namrata, Ishita, am I audible to all?
Yes,
Am I
sir.
audible?
Yes, sir.
Yes,
Sir.
yes.
Okay, so please. Okay, but your voice is not coming to me correctly. So please speak again.
Sir, it is a set of gene which is transcribed under the control of an operator gene.
Okay, so simply it's not perfectly fine definition. So it's a set of gene which operate under with a single switch, you can say, which operate together, which you required for the transcription. Okay, so as you have studied the lac operon. So can you tell me the component of the lac operon? Can you tell me the component of lac operon?
So,
Sir,
regulatory gene, structural genes,
Shrita galactosides.
and operator.
Yes, Shifta, please
So you
speak
said
again.
components or the components of structural gene. Is that
I'm
right?
asking component of the lac operon.
Sir, regulatory gene, operator, and the structural gene.
Okay, so what are the regulatory genes? So simply, I can say you that when you're talking about the expression of a gene, you need all the machinery that you required for the transcription. So the machinery that required for the transcription simply means you need a RNA polymerase gene, RNA polymerase you need. So for the RNA polymerase, you have to recognize the sequence, means it have to bind to the location to the gene. For the binding location of the gene is called as a promoter, so means have to bind a site that is upstream to the gene, and when your RNA polymerase is going to bind to the promoter, then it will start the transcription. So when it starts the transcription, then you need at the end point where your transcription should stop. That's called the terminator. Then again, you need on your gene that mRNA that you are going to form. On mRNA, you also need one more site that is called the ribosome binding site. So ribosome binding site is going to determine whether that from after the transcription translation should occur or not. So these are the things that you need with your gene. But when we are cloning, at the time of the cloning, we do not bother about the promoter, terminator, or the ribosome binding site. Then from where these are going to come? So these sites, these things should be come from your vector itself. So now from our vector, earlier we mentioned that we are introducing your restriction, multiple cloning site. We are incorporating your markers, also the origin of replication. Now we need an additional feature from your vector that should help us also in the transcription and followed by the translation. So means what you need. So you have to incorporate one thing. What is the one thing that is a promoter? So what is promoter is? Promoter is a location. Promoter is a site present on your gene which help in the binding of your uh, RNA polymerase. So it is going to be upstream, it is going to be before your gene. So this is a promoter site where it is going to bind. So now here at this promoter site, who is going to bind? It is going to bind your RNA polymerase. So simply you can say that this is the site from where your transcription will start. So transcription will start from the your promoter site. So the next sequence to your promoter will start will be part of your mRNA. Okay. So now what you need next? Next you need is from this site your mRNA will start forming, but you need in your mRNA one more site that is going to be a ribosome binding site. This ribosome binding site should be before your start point of your gene. Start point of your gene, that means it is going to be called as a start codon. So before the start codon, you need one more thing that is called as the ribosome binding site. This ribosome binding site is the recognition site which your ribosome, that is your, as we are talking about in case of your prokaryote and your prokaryotic ribosome, that is your 70s ribosome, should recognize it. Otherwise, your 70s is not recognizing it, then it won't translate. And if you are cloning into eukaryotic system, then your 80s ribosome should recognize it. Otherwise, it won't go for the translation. So first thing you need a promoter, which is going to be a site where your uh, RNA polymerase will go and bind and start the transcription. So now what type of the promoter you use, and depending on what you need, and so all that on that basis we design our promoter, we select our promoter. So main job of the promoter is going to be your transcription start. So once the transcription is going on, so you need a site which your RNA polymerase should read and stop the transcription. So that is going to be your terminator. So this promoter, that is, as you know, in case of your, if you remember, in case of your lac operon, so lac operon is going to have promoter, then operator, then your structure gene, then followed by the your terminator. So these are the components as of your lac operon. So all the components are going to be required here also when you want to go for a transcription and followed by the translation. So when you transcribe it, so promoter, from that site of promoter your RNA polymerase will bind, and this is the start site from where your transcription is going to occur, and whole the sequence is going to be there in your RNA. So and at the point of terminator, your RNA polymerase will stop the transcription. So all these components you need. So now we need to know what are the component, whom you are going to call it, and what are the sequences, what are the feature of your promoters. So as we mentioned, the promoter is going to be upstream to your start site. Start site means upstream to your start codon. So how much upstream? As you remember, your lac operon and trip operon, we mentioned that it is going to be -10 to -35 upstream to your start site of your gene is your promoter sequence. What are the promoter sequence? If you remember, same, nothing is new here. It is going to be Tata box or Prevnar box. So these are the TAAT, that is the -10 to the gene that's located in your E. coli, and -35 it is going to be located at TTGACA. To this we call the Prevnar box. So if Tata box or the Prevnar box, we are going to call it as a promoter sequences. And in between these -35 to the -10, some other sequence may be there, but these are the conserved sequence. So they remain same, or simply, or you can call it the consensus sequence. So they may have a minor variation, but most of your cells are going to have this sequence. Okay. But in case of higher organisms, so this sequence may vary. So as you can see that at the instead of -10, in case of your eukaryote or higher, you have at the -25. Okay, so this is a site which your RNA polymerase is going to recognize. To this we call it the promoter sequence. So it means what you need to do. So if depending upon what expression system you want to incorporate, you have to incorporate these sequences also. Okay, so these as here the written sequences are of E. coli and also some of the animals, but each animal is going to have minor variation in these sequences. So on that basis, we classify that this is a promoter of that organism or another organism. So one more variation are there with the in between sequence what we are referring between the -35 and the -10. So still you can see 50 nucleotides are here also. So what they are, they also vary, and on that basis you describe that this is of that. So genes of higher organisms are also surrounded by expression signals. Their nucleotide sequence are not as same of the E. coli as we already mentioned. By comparing the promoter of E. coli and human as seen below, there are similarities and unlikely that E. coli RNA polymerase will be able to attach the human promoter because they are not. There are some similarity, but dissimilarity is also there. So that's why two polymerase they can't recognize each other's promoter. So what we are referring here, we are referring a foreign gene. Foreign means we are cloning into the E. coli. So in that case, what you need a promoter which should be recognized by polymerase of your bacteria. Okay, so because bacterium does not recognize its expression signal, okay, so that's why you need a promoter of that. Otherwise, your gene is going to remain inactive within your E. coli, even if you have taken with your gene the promoter sequence of your foreign gene. So in that case, what you need a bacterial origin, or if you need a sequence which is recognized by the RNA polymerase, which is present in your bacteria. Okay, so how we are solving it? So we are solving to insert foreign gene into vector in such a way that it is placed under control of a set of E. coli expression signal. So in your vector, we are introducing E. coli expression signals also, and we are cloning downstream to this expression system your gene. If you are cloning downstream to these signals, then your gene will serve as a your vector will serve as an expression vector, and your gene will be going to be expressed also. Okay, so it can be achieved. Then gene should be transcribed and translated. Cloning vector that provided these signals are able to express, able to produce these recombinant proteins, and such vectors are called as a expression vectors. Okay, so these are the vectors which contain the expression signals. So expression signal means upstream should have a promoter and ribosome recognition site, and downstream it should have terminator sequence. So upstream and downstream is upstream means before your gene, downstream means after your gene. So from here, how we are going to get? So simply you can say that the E. coli expression signals, promoter you need, ribosome binding site you need, and a terminator you need, and unique expression site should be after the ribosome binding site. So what you are going to do? In between you are inserting your gene, you are cloning, in between these your foreign gene, and now this foreign gene, if you transform to the E. coli, so under E. coli expression system, transcription system, or translation system, it is going to transcribe and followed by translation. Means you are going to get your protein back. Okay, so now coming about the promoters. What different types of the promoters are there, and why these are so important when you are trying to the expression. So these are the backbone of your expression. Means these are the backbone. Means if they are there, then they are going to facilitate the transcription. So as in case of as we mentioned that in case of our cloning vector, they should have high copy number. So here also these promoters should be like that. They keep on means when they should be like that. When you need, they should express. When you don't need, they should not express. It simply means when you need, then the RNA polymerase will go and bind them, and when you don't need, then RNA polymerase won't go and bind there. Or with that, they should produce a large number of mRNA molecule. If they are producing a large number of mRNA molecule, it means in return, these large number of mRNA molecule will translate and produce large amount of your protein. Because the main aim of our generating this expression vector is to get recombinant protein in a large amount. So your promoter are going to affect them at a great extent. So we choose mass them with care. So sequence are consent sequence. Average of all E. coli promoter sequence are taken that are known. Although most of E. coli promoter do not differ much. See, I do not as from these consent sequence. Example, TTTACC is there. In some, it is going to be TTGACA. So these are that's why we are saying they are conserved. They are not conserved. They are consensus sequence. Minor variation may be there, but this small variation may have a great effect in that expression also. So that's why it is must how to use what care we should keep while designing our vector, what promoter sequence we must incorporate. Okay. So now one more point is there about is that the strong promoter and the weak promoter. Strong means which is going to express your gene strongly, means which is going to transcribe much more. As it is simply you can say it is just as about your copy number. So promoter which give multiple copy of your mRNA. To that we are going to call it as a strong promoter. Weak promoter is going to give you less copy of your mRNA. That is going to be your weak promoter. So thus, strong promoter that can sustain a high rate of transcription, we call for it as a strong promoter, usually control genes whose translation product are required in large amount. So where you need a large amount, in that case we need a strong promoter. But sometime what happens, sometime when you produce your recombinant protein in a large amount, that protein may be toxic to your cells also. So in that case, always we don't need a strong promoter. Sometimes we need a weak promoter. So weak promoter means which is going to express which is going to transcribe in a lesser amount. So when it is going to transcribe lesser amount, then it is going to produce a lesser amount of protein. So here you can see this is strong promoter. Strong promoter is producing a large number of mRNA, and large number of mRNA is going to produce a large amount of your protein in a single round. Wherever in case of your uh, so wherever in case of weak promoter, it is going to transcribe less number of RNA and it is going to produce relatively low amount of your protein. So this is all depend upon what your utility is, whether you need a strong promoter or you need a weak promoter. So both type of promoter can be there, strong and the weak. So now coming about different types of the promoter that you are using in your expression vectors. So as I mentioned, these promoters are the part of your operons. So different operons have different operators, so different promoters. So as you know, the lac operon have a lac promoter, tryptophan operon have a trip promoter, tag promoter, lambda PL promoter, and the T7 promoter. So these are the different promoters which you get from the different operons and which you are going to utilize in, while developing your expression vector. So each of the promoter have its merits, also it is going to have its demerits. So now come to just some information about these promoters. So first the lac promoter, as the lac name, the lac is there. So from lac operon you are going to get it. So what it controlled, how it being controlled, control the transcription of LAC-G genes that code for the beta galactosidase. Also, LAC-G fragment carried by the PUC M13MP vector induced by the. So these promoter is going to present in your all these PUC vector M13MP vectors. And how it works? It works by the presence of IPTG. isopropyl thiogalactosidase. So it is going to be a substrate. So if this substrate is going to be present, then your promoter is going to work. Otherwise, this promoter won't work. So this is an inducible operon, you can say, inducible promoter. So when you add LAC-G genes, then promoter will express. Otherwise, this LAC-G genes won't work. So how this LAC-G is going to work? How this promoter is going to work? This IPTG as serve as a substrate. So this promoter play a role in the production of your RNA polymerase, which is going to recognize this promoter sequence. Addition of this chemical growth medium switches the transcription of gene instead of a downstream inserted downstream lag promoter carried out by the expression vector. So this is a simple promoter that you are using in your PUC also, as we already discussed about this lag genes and from where this lag promoter is being taken up. Next one is the trip promoter. So tryptophan, you know that is a respiratory system. If you add the tryptophan, then this promoter stop functioning. So normally, so where it is present, normally upstream of the cluster gene, you have to add it coding for several of your enzyme involved in the biosynthesis of amino acid tryptophan. So this is a source of your trip promoter. How it works? It works in a repressive way. So if tryptophan is going to be there, then this promoter won't work. If tryptophan is absent, then this promoter is going to work. Okay, so how it will be induced? So it is to be induced by the 3B indole acrylic acid. So in presence of it is going to be induced. So if this is going to be there, then it will express. If tryptophan is there, there is going to be no expression. So simply how it is going to work, this is going to be your next type of your promoter. So liquid like tryptophan promoter is there. So then you after this you are cloning your gene. If you are adding to your system 3B indole acrylic acid, then transcription is going to be there. If you add the tryptophan, then no transcription is going to be there. Whereas in case of previously it was inducible one. So when you add the IPTG, that is substrate so that is isopropyl thiogalactosidase. So if you add it, then going to be transcription. Otherwise, there is going to be no transcription. So you can control your. So when you need expression, you can use it. When you don't need expression, then you don't need it. So next type is going to be your tag promoter. So what is tag? This is a tag. It's a mixture of lag and trip. So T from the tryptophan and ag from the lag. So it is a hybrid of it. So hybrid is going to be work in a way that it's going to utilize by the. It become more or less it is inducible. It is not a repressible one. So when you hybridize both the sequence, then you got a stronger one. Like it is a bit stronger than both tryptophan and the lag. So this is going to be your tag promoter. It is also inducible one. How to induce it? You can induce it by the IPTG. So when you add IPTG to your culture medium, then your gene will express. Otherwise, your gene won't express. Then the next type of promoter is going to be lambda PL. So here the lambda means it is going to be originated from the bacteriophage. So promoter responsible for transcription of the fast DNA molecule. So when the fast DNA molecule is within your cell, then it go for the transcription of that DNA molecule. So it is again a very strong promoter. So what it recognized by it? It is recognized by the E. coli RNA polymerase, which is subverted by the lambda into transcribing bacteriophage DNA. So this promoter is also recognized by the E. coli RNA polymerase. You don't need RNA polymerase. You don't need to modify your host for the RNA polymerase. Just you can utilize the RNA polymerase of your host E. coli. Okay, so the promoter is repressed. It is repressible one. How can you repress it? By the product of lambda CIG. So lambda CIG, if the product is there, then it will be repressed. Otherwise, it will be. It is going to be. It is all. It is. You need not to use it. So you keep on expressing it. So expression vector that carry lambda PL promoter are used with the mutant E. coli host that synthesize temperature sensitive form of CI protein. So what you are going to use? Here you are going to use a mutant E. coli. This mutant E. coli is a temperature sensitive, means when you want to suppress the activity, then you can raise the temperature. So temperature, on the temperature based, it secretes the CI protein. Okay, so here how we are going to work with it. So at low temperature, less than 30, mutant CI protein is able to repress LPL promoter. Higher temperature, protein got inactivated, resulting in the transcription of your clone gene. So if you keep your E. coli below 30 degrees Celsius, then this CI protein is active, okay, and it will stop the transcription. But as you rise the temperature above 30 degrees Celsius, CI protein will become inactive and transcription will start. So this is how different promoters are controlling the transcription of your gene. Now the next type is a T7 promoter. So T7 is a bacteriophage. It's unlike lambda-phage. So what it do? So T7 bacteriophage have its own RNA polymerase, specific RNA polymerase coded by T7 bacteriophage. So now you need to note one thing: this promoter need RNA polymerase coded by the T7 bacteriophage. Means if you are using this promoter, then your cell must have RNA polymerase that is of a T7 bacteriophage. Okay, otherwise this RNA polymerase, this promoter won't work. When even in your cloning vector, if this promoter is going to be there, whether you express it or not, uh, whether you add any type of the IPTG or so on, you are not going to get any expression. So what you need? You need a RNA polymerase of T7 bacteriophage. So what do we need in this case? In that case, we clone or we insert. So double cloning is there. Here we are modifying our host itself. So what you are doing here? RNA polymerase much more active than the E. coli RNA polymerase. So this is the beauty of this RNA polymerase, that its activity is faster than host RNA polymerase, which means the gene inserted downstream to T7 promoter will express at a high level. So it is further a stronger, this promoter is stronger, with that RNA polymerase also stronger in this case. Okay, so gene of T7 is not normally present in E. coli. So as I mentioned, it is not there in the E. coli. So what you need a special strain, which strain have this T7 uh, bacteriophage RNA polymerase. So how you can get it? So for that we are having a lysogenic T7 phage E. coli. So what we did? We infect E. coli with T7 bacteriophage, and this T7 bacteriophage got inserted in the genome of your E. coli. When it got inserted in the genome of E. coli, means gene of your T7 bacteriophage are present in the E. coli. Okay, so now lysogen contain the inserted copy of phage DNA in its genome, means your E. coli having the phage DNA. When the E. coli having the phage DNA, then it is going to produce T7 RNA polymerase. So this T7 RNA polymerase is now here is a some bit complicated thing is there. So now what we are having, we are talking about the T7, we are talking about the promoters, but now we start talking about the RNA polymerase also, because this RNA polymerase, when we are using T7 promoter, so this promoter is going to be present upstream to your gene. Means we are cloning. What we are cloning? We are cloning gene upstream to what? Upstream to our vector. So, but this T7 RNA polymerase is present in E. coli. E. coli genome. Until unless T7 polymerase is not there, means until unless this E. coli won't express this T7 RNA polymerase, it won't start the transcription of your foreign gene. So we are indirectly controlling. Now we are not controlling the we are not control we are not controlling binding of RNA polymerase at this site. But what we are doing, we are controlling the expression of this T7 polymerase. This T7 polymerase lack promoter. So we are what we are having. We are having dual thing. In gene, we clone, we transfected T7 polymerase under the control of Lac-Operon is going to, when the Lac-Operon is active, then T7 polymerase will form. And when this T7 polymerase will form, this T7 polymerase will go to your vector and will start the transcription of your gene. Okay. So lead to the activation of gene carried by the T7 expression vector. Okay, so these are your expression system, your expression vectors that we use. This simply means what are the features are there in your cloning vector. Other than that cloning vector, you are going to have promoter upstream to your gene, and also ribosome binding site upstream gene, and terminator sequence downstream to your gene. Okay, so that will help you in the expression. So just wait for a minute. Okay, students, got this? Okay, so this was a feature of the expression vector, and with this we ended our module number 3, your vectors. In it we talked about all the vector, that your expression cloning vector, plasmid vector, fast-based vector, large cloning vectors, yak and back, and the expression vectors. Okay, so this is all about your vectors, and with this everything is done. If any one of you have any questions regarding expression vector or today's lecture, please ask. Any question? Amrita, Manaswini,
No, sir, no questions.
everything is clear?
Yes, sir, it's clear.
Okay, so now we have done, so still we have few minutes, so if anyone have any query, you may ask. Okay, so if no questions, then please turn on your camera while call your name. So one more thing, I will, those who have joined late means after 12:25, they are absent. So multiple time I have told you that join on time, but still continuously many of you are joining late. So I have extracted the attendance list, and I will use that attendance list. Those who have joined before 12:25, I will mark them present. Otherwise, those who joined late, them I will mark absent. Okay. So please turn on camera while I call your name. Adesh.
Present, sir.
Atoiba. Demetrius.
Present, sir.
Devanshi.
Yes, sir.
Ishita.
Present, sir.
Kshitij. Manaswini.
Present, sir.
Namrita.
Present, sir.
Nandini.
Present,
Nandini.
sir.
Nehal.
Present, sir.
Rupal.
Present, sir.
Saiba.
Present, sir.
Sanjana.
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir. Present, sir.
Vivek, 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 Yukta,
Present, sir.
Yuvraj.
Present, sir.
Okay, so those who are absent, just Kshitij is absent today, and we have 2
Present.
more minutes. So, Kshitij, you are absent. At what time you joined today?
After 12:25, but
It's regular. You are doing it every day. Okay, so today you should get some penalty. So otherwise, you won't come on time. Okay, so meanwhile, I can evaluate Rupal's paper. Okay, so Rupal, are you able to see your screen? See your paper? So let me determine the concentration. Okay, so in this question you didn't write the principle. So in the principle you have to write about the Beer-Lemon law. Molecule absorb different wavelength of light, various degrees. For estimation, quantification. For the quantification you didn't write. For the quantification you should write about the 260 nanometer and also what OD you are going to get. After that, if the absorbance is going to be 1, and for quantity evaluate by the ratio. So ratio, yes, but what this ratio means, high or low. So very poorly attempted question. So just 1 mark for this. Reagent used in DNA extraction, lysis buffer. So what this lysis buffer composed of? Chloroform used to increase the efficiency of phenol to denature protein. Chilled protein used to precipitate. D buffer. So what are the components of pre-sanitary DTA? Phenol decrease denature proteins. Tris maintain the pH. Okay, so it is also very poorly attempted. 2 for this. The plasmid extraction, Tris HCl. Lysis buffer contain STS, disrupt the cell wall. Do not disrupt the cell wall, disrupt the cell membrane. Neutralization buffer contain the acetic acid. So what is the role of promote renaturation of plasmid DNA? Yes. And what the lysis? It also contain one more component, NOH. We didn't write that one. Okay, so this thing. Sealed propanol precipitate the seed. So 2.5 for this. Rupal, keep on counting. Okay, next is gel electrophoresis. Depend upon the matrix, shape size, same DNA differently. It is not, it's a 0 for it. You have to write size is same, means that is going to be the size, charge, everything are going to be same. The flexibility of steric hindrance of your molecule, you have to write about it. So nothing for this. Resting endonuclease capable of cleaving side sequence. So altered lex Okay, fine. So why it is doing so? You write it. Keep in mind, DNA should be free from no store. No, 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 no. One for it. So how is the total, Rupal? Rupal.
Yes, sir.
So कितना हुआ total?
I think 6.5.
1, 2.5, 3.5, and how much for this? I think 5.5. Rupal, you want to defend? Do you think somewhere you should get more marks? You can ask.
Sir, it's okay.
These are final
It's fine,
marks.
sir.
These are going to be final marks, and they are going to be the part of your end term exam also. They are part of your condition assessment. Okay.
Yes, sir, I know.
So कहीं लग रहा है कोई गुंजाइश है तुम्हें लगने की कि यहाँ पे मिलना चाहिए, इसका नहीं दिया मैंने मार्क्स, बता सकते हो?
Sir, in gel electrophoresis, the gel electrophoresis question.
In the gel electrophoresis question was on the gel electrophoresis, on the agrogel electrophoresis, DNA molecule of same size may migrate at different speed. So why? So gel matrix act as a CU small, so all are of same size. Different conformations are there, they migrate differently. Why? This was a question. Different form of same size DNA run differently. Agarose gel fragment negative. So when the size is same, negative charge is going to be same. No change in negative charge. Everything is same. So why they are migrating differently? That you have to answer. For that, I showed you different. That's image also, slides also on that. That supercoiled migrate at different speed. Then your open circular migrate at a slower one. Open circular slowest one. Supercoiled faster, and linear even faster. So what make them to run faster? Okay, so on that day I showed that what make linear to run faster and what make that you have to write in this question. So that's why this answer is in no way near to the answer. Okay, in the star activity I give you the marks. So I ask you a star activity you have to write why this is going to occur, why they are recognizing a different restriction site. And also in this one you should write what condition you should keep. So buffer condition, the pH condition, so that you should be you should take care of them. These precautions that you have written, these are common for whether you are doing double digestion or single digestion. Okay, got it, Rupal?
Yes, sir.
So these marks are of just this test. So they are not out of 45. So 20 marks are still remaining. They are there from your Viva and your lab report. Okay, so Atoiba is also here. Atoiba? Atoiba?
Hello.
Okay, so here is your test. So principle of spectroscopy, nucleic acid, running sample through egos. So in the spectro photometer के अंदर कहाँ से आ गया भाई जेल? Wavelength. Atoiba 0 for this. Question number 3. Alkaline lysis method used to make the cell. Suspension buffer contain trace HCl and buffering and maintain the okay. RNAs to delete DNA. RNA. Lysis buffer contain STS. Disrupt. It is cell membrane, not the wall. It's not NaCl. It's NaOH. Utilization buffer contain acid, potassium acetate. I'm trying to promote the renaturation of plasmid DNA. Yes, this is fine. So for it, I'm giving you 2.5. Star activity for the laxation, alteration, occur reaction condition that differ significantly. Okay, so non-recognition site, that's fine. That's it. This is half, so 2.5 for this. 2.5, 2.55. That's it. Atoiba? Atoiba?
Yeah.
So 5 for you. Okay. So now our time also over. So thank you all. Stay safe, stay happy.
Thank you, sir.
Thank you, sir.
Thank you, sir.